Last time we started with DNA structure and function presentation, and before we continue on with transcription and translation, I just want to give a short review about what we covered last time. So bringing you back to this slide where we want to orient ourselves as to the stages we are at when we make a new cell. We have talked about the transport of nutrients into the cell. We've talked about the breaking down of nutrients into smaller building blocks through metabolism and how we extract energy from those nutrients. And now we want to take those micromolecules, those building blocks, and we want to make new DNA, and we also want to make proteins for that new cell. And so we started off talking about how we can replicate that DNA. That new cell needs the exact same components that the old cell needs, and we started off with replication of the DNA. And so this next part of the presentation will be looking at how we can get RNA from DNA through transcription, and then how we use those different types of RNA to get proteins through translation. So just a reminder that our genomes are all genetic material contained within an organism. And depending on the type of cell, we have different types of DNA. And so we went through the different types of DNA that you'll find in a prokaryotic cell versus a eukaryotic cell. Eukaryotic cells being either animal or plant cells, and also what viral genomes consist of, DNA or RNA, but not both. Then I spoke about genes, chromosomes, how those chromosomes are divided into genes, because it's the genes that are the fundamental unit of heredity. And they're going to code for different traits, cell functions, and also code for RNA that we can use to translate into proteins. And so these different categories of genes, we'll be looking at structural genes that do code for the proteins, genes that just code for RNA that are used in protein production, and then those regulatory genes that are going to control the expression of those genes, either turn genes off or turn them on. Also to remind you that the DNA double helix, the structure of the DNA, can be referred to as a ladder, with the handrails being the sugars and the phosphate groups joined together along these ribbon-like structures. And then the stair steps of the ladder are the bases that are joined together by hydrogen bonds. The bases of the DNA being the adenine, thymine, guanine, and cytosine. And guanine always pairing with cytosine by by three hydrogen bonds, and the adenine always pairing with the thymine with two hydrogen bonds. And then there's this concept that I mentioned about DNA replication being both complementary and semi-conservative. And so the complementary part of the DNA replication is just basically that when DNA is replicated, you have the adenines pairing with the thymines and the guanines pairing with the cytosines to produce these the double-stranded DNA. And then you have this concept of DNA replication being semi-conservative, where the old strands are separated before replication, and then the new strands are using the old strands as a template. And at the end of DNA replication, we're going to end up with two cells having one old strand of DNA and one new strand of DNA. And so in that case, it's semi-conservative. So now that we have our DNA replicated, we can also use that DNA to transcribe and translate proteins. These two processes together, transcription and translation, equal gene expression. And so we're going to look at how do the cells take DNA to RNA and then take that RNA to translate it into proteins. With that, I'll remind you about the central dogma of molecular biology. And what this means is that information is flowing in one direction, from DNA to RNA to proteins. There are some exceptions where RNA can be taken back and reverse transcribed into DNA, but for our purposes, we're just going to look at how we can use DNA to get RNA and use that RNA to make proteins that we need for the new cell. So remember that DNA is going to encode all the information needed for those molecular components that we need for replication of new strands of DNA and also to produce a large number of proteins that the cell needs. The process to get from DNA to RNA is called transcription, and the process that we use to take RNA to proteins along with amino acids is called translation, and we'll see how that works. So, a reminder of the structure of proteins. We've seen this figure a bunch of times during the semester, and just to remind you that the primary protein structure is just the sequence of a chain of amino acids. So remember these little circles look like beads on a string. Those are the amino acids that we can piece together to make proteins. Once we have our primary structure, then we can start folding. Those amino acids are going to interact with each other to make alpha helices or beta sheets. Those alpha helices and beta sheets can further fold on themselves to create this tertiary structure. And remember, this tertiary structure is that 3D globular structure that you'll find in proteins. And then some proteins will also form this quaternary structure where you have some proteins that consist of more than one subunit of amino acids. When proteins are translated, they consist of 20 different amino acids. There's 20 different amino acids that are used in all forms of life, and they are joined together in different sequences and arrangements to make hundreds of thousands of different proteins. You can see here the beginning of a protein that starts with the amino acid glycine. These amino acids are linked together in synthesis reactions in order to form these long strands of primary structures. This table shows you the 20 different amino acids, the long name and the 3-letter abbreviation for each amino acid. And so this will be helpful when we actually get into translating proteins from the RNA. The master genetic code. The master genetic code is going to be that representation of amino acids. The DNA code that is contained in our genes is going to be the overall determinator of the order of the amino acids in the growing protein string of beads. And so each amino acid that comes next in that growing chain is going to be represented by a code on the RNA that's called the codon. And it's this codon that consists of a set of 3 RNA nucleotides. And so for every 3 nucleotides on the RNA, you have a genetic code for a single amino acid. And so we'll see later how we can use this table here to determine what amino acid is going to come next in our growing chain of protein. And then how do we convert the DNA nucleotide language into that codon language that we need to determine those amino